Good morning, everybody.
Good
All
morning.
right, that looks like a nice spot. Okay, so I think we finished off the brief overview of the test cross. My memory serves me correctly. Come on, get you up there. All right, so chat back. All right, let's rock and roll. Here we go. So I'm pretty sure we finished up here. I think I checked the YouTube video that I uploaded from last Thursday. And I believe that's where we ended up. Okay, so just as a recap, we've gone through our monohybrid cross stuff. All right, and so again, remember there are 3 different stages, generations. They each have a very specific makeup in terms of genotypes. And also always remember or think about what alleles, pardon me, or rather what gametes can you get from each parent in that generation, right? That's really the key to understanding how to do these types of crosses. Because as soon as you get that, it's straightforward. You just add them together and then you get the next generation. Okay, so keep that in mind. We're going to wander over to the whiteboard, which I just cleaned. It's like spotless. It is absolutely beautiful. That's also why I was a little bit late. Got a little bit carried away. Just so excited about having a nice clean. I don't know why that's kind of up there. My computer's acting all kind of crazy. So if I look like I'm speaking slowly, it's not because I'm being really slow. It's because my computer's doing something funky. All right. Okay, so let's wander over here. Find somewhere to put my coffee. You can see all my super cool stuffed toys. I'm going to try sitting down just because I'm a lazy ass and I don't want to stand up. But if I need to, I've got plenty of space. Okay, so essentially the issue that we have is the presence of one dominant allele gives you the dominant phenotype, right? So essentially, let's use A. Actually, an or is better there. Oops. It's a little trickier because I've kind of got to use both hands here. Equals the dominant phenotype. And I'll take pictures of this as we go. So if you have an individual with the dominant phenotype, what's its genotype? No way of knowing, right? That's where the test cross comes in. So if you want to figure out for your dominant individual, could be a dog that's supposed to be pure breeding, or chicken. People go nuts over chickens. Like my daughter actually always wants chickens. Can I have chickens? No. Can I have chickens? No. Anyway, you can have a snake. No, actually, you can't have one of those either because my wife doesn't like mousicles in the freezer. So the way to get at this is with a test cross. Now, what genotype, or even phenotype actually, it's kind of clue there. What genotype would you have to cross this unknown individual with, or this dominant individual with, to see whether it's homozygous dominant or heterozygous? You're going to do a cross, right? So you need another parent essentially, to cross this unknown with, right? What would the genotype? Wow, holy crap, baloney, that was quick. Didn't even finish speaking. All right, yes, homozygous recessive, because that's the only way you'll see. That's a crappy arrow. There you go. If you have a recessive allele present. So, and you can try, you can like write it out with other genotypes and you'll see it just doesn't kind of really work. Also, there's the issue that you know what the genotype of a recessive individual is, right? Because the only way it can be a recessive phenotype is to be homozygous recessive genotype. So you know exactly what the genotype of that individual is, which makes it a very good tester, right? There's no uncertainty. So essentially what you have, and this is how I always write it. Sometimes it works for some people, sometimes it doesn't. So if it doesn't make sense, give me a shout and we'll do it a different way. So this I like to call the tester, right? So this is, can you read this okay? It's a little bit of an angle just because I'm sitting down. There's also, my head isn't like super large. I just have to like my laptop in my other hand. Thankfully, it's a light one. Anyway, so this is our tester. And essentially, we're going to cross our unknown with this tester and look at the progeny. This is not a monohybrid cross. Monohybrid cross is always P1 with homozygous dominant, homozygous recessive, heterozygous F1, and a 3-to-1 ratio in the F2. This is not that, right? Because as you can see, you don't know what the other genotype is. So we have, I don't know, question mark number 1. Yeah, bloody lights. Let me turn the lights down a little bit. You know, these fancy, oh no, we don't have these fancy lights. Ah, well, that'll do. I was going to say, we've got these fancy lights, which we can, alter the level on, but I can't because they're too fancy for me. I can't figure out how to use them. It took me like 3 months to figure out how to turn the lights on in here. So anyway, actually, let's put that down a little bit. My whiteboard is too clean. So let's say the unknown is homozygous dominant. First step always, like Matt's patented success at Mendelian genetics plan, what are the gametes that you're going to get from these two individuals? What alleles will they have? What gamete or gametes are we going to get from the homozygous dominant individual? Are unknown. I'm wagging my eyebrows at you. What is the only allele that we have here? Yeah, it's a large A. It's like you can have any color as long as it's black kind of deal. Right? And this seems like a little bit laborious in a sense, right? Doing it this way, but this will help you a great deal when we come to dihybrid crosses because things get kind of more complicated. And the same for here. What's the only gamete you can get from this individual? Yeah, exactly. It's going to be a recessive allele, right? And so you combine those together via fertilization, and you get only heterozygous individuals, much the same as you do in the F1 generation of a monohybrid cross. So 100% dominant. Right, that's pretty straightforward. Now, sip of coffee. You like my coffee cup? Nematode whisperer. How cool is that? Anyway, for our second unknown, unknown number 2, right? And again, it could be either of these two. What we're looking at is the offspring, the ratios particularly. Now, instead of being this cross, now we're going to do this cross, right? So what gametes can we get from this individual? This is very much an example of a test cross, Clarissa. Yeah, this is how essentially the nuts and bolts of it. Excellent, Michael. Yes, it's either going to be dominant A or a recessive A. Now, according to the law of segregation, right, which states that which allele a gamete gets is random, if there are two different alleles, what is the ratio of these gametes? Like how many of these dominant allele gametes are we going to get to recessive allele gametes? Yeah, I can lip read, Jess. Good job. It's going to be a 1-to-1 ratio. So, and you can see where I'm going with this already, right? So you're either going to get, again, heterozygotes, or I think my neighbor's playing some nice music, or homozygous recessive. So now we come back over here. Now you can see this recessive allele, right? It becomes apparent because now it's going to be joined with one of the recessive alleles from the tester, right? And because we have a 1-to-1 ratio of gametes, then you're going to have 50% dominant, 50% recessive. So if you ever see, and there will be questions on this in the exam, guaranteed, at least, I can't remember how many, like 2 or 3, maybe. You're going to be asked something along the lines of, we have, I don't know, what would be a good example? Kind of a flippant one. We have a beautiful donkey crossed with a scraggly donkey. Best I could do, sorry. And you get a 50-50 ratio. Let's say you get like 3, what did I say? Beautiful donkey? Oh, I can't remember. And 2 scraggly donkeys. Or let me say 4 scraggly donkeys out. What's the genotype of the beautiful donkey? It's going to be heterozygous, right? Because that's the only way you're ever going to get the recessive phenotype appear in the progeny of the test cross. And so there's 2 kind of clues, essentially. One is the ratio, right? And so it's either going to be 100% dominant, 100 or 50-50, right? If it's 50-50, it's a test cross. Cannot be a monohybrid cross because there's no way you can get a 50-50 ratio in a monohybrid cross in any part of it. The other clue will be the structure of the cross, right? So a monohybrid cross has 2 different crosses, essentially. One for the parentals and one selfing cross for the F1 generation. So if I'm just doing one cross and looking at the progeny, it's not a monohybrid cross. It's got to be a test cross. right? So you can kind of come at it different ways, but either of those will kind of go, wah, wah, wah. We're dealing with a bad test cross here. And as soon as you know you're looking at a test cross, then you go, oh, okay. What we know about test crosses? There we go. That's what that is, right? So that's kind of partly your understanding of what we're doing, but partly an exam skill, right? What are the key bits of information you're looking for, right? Are we talking about crossing heterozygotes together and getting a 3-to-1 ratio? That's a monohybrid cross. We're talking about crossing unknown to a recessive phenotype and getting a certain ratio or not. That's a test cross. Okay, so kind of I give you all the information that you need to answer the question in the exam. I'm not trying to trick you or hide stuff, but you have to know what you're looking for. All right, let me take a picture of that while you have a think. And ask any questions if you have any. Wheels on chairs is like the best thing ever as far as I'm concerned. It's funny, I've spread out my board work so much just because I have so much space. And also don't have to do with my dog lying down in front of it either, which, although very cute, is a bit of a pain. Okay, anybody got any questions about test crosses? Nope. Cool. If any do come to you, just give me a shout. Bloody hell, this blue is such a pain to get off. All right, doing everything in red from now on. Get some nice lens cleaner on my coffee. Okay, so this is a kind of like useful skill or thing to be able to use. And really, in many ways, it's something that's used, even if they don't realize it, by a lot of people who do breeding, right? Dog breeders, for example, crop breeders, although now we have a lot more kind of tools with which to know what we're dealing with genetically. Actually, in many ways, now you just sequence the animal, but easy peasy, right? You don't have to do a cross or anything. But this is still a valid technique for figuring out what you have. Okay, so now we're going to get on to, we're making pretty good progress here. Oh yeah, I love my sleepy puppy too. It's actually pretty kind of special to have, you know, dogs lying at your feet while you're teaching. I think that's like totally awesome. All right, so what we're going to do now is the dihybrid cross, right? And so let's just get this up. Okay, so the deal here is that this is exactly the same as a monohybrid cross, but now we're dealing with two traits. So monohybrid is one trait, dihybrid is two traits, whatever those might be. I've used all kinds of silly examples in the past, right? So now for each of those traits, we have two different alleles and two different phenotypes. So in the parents, we have, they're going to be homozygous. Like, as you know, for the monohybrid cross, the parents are always homozygous, either dominant or recessive. But now we have two different genes to work with and two different phenotypes. So far, so simple, right? So now we have seed shape and seed color, for example. And so the phenotypes for those are round and wrinkled, and yellow and green. So if you just kind of put your hand up to the screen and cover up the genotype, here's a question. How would you figure out what are the dominant phenotypes? If I didn't give you the genotypes, how would you figure out what the dominant phenotype is? What would you look at? Because you can't tell by looking at the, yeah, no, not good idea, Odalise. Not the ratio. I think you're thinking of the F2 generation. You're right in that you need to look at a generation, but which generation are you going to be looking at? Not the F2. Well, actually, yeah. I mean, yeah, you could use the F2. The F1 generation, right? Because what is the genotype of the F1s? Always. If you're crossing a homozygous dominant to a homozygous recessive, what's the only genotype you're going to get? Genotype, Mika, not phenotype just yet. What is the genotype of the F1 generation? Not dominant, that's a phenotype. Just on its own, that refers to a phenotype. Heterozygous, yeah, exactly, Jonathan and Amanda. It's going to be heterozygous. Doesn't matter how many genes you're looking at. You could do a heptahybrid cross, which I think is 7. Never done that. I don't have a big enough whiteboard. But if you're saying, I'm going to do a heptahybrid cross because I'm nuts and I've got nothing else to do, right? You will have exactly the same principles, right? You'll have 7 genes, and well, I don't have enough fingers on that hand. 7 genes and 7 genes. One individual, one parent will be homozygous dominant for a particular gene. The other one will be homozygous recessive. The F1s will always be heterozygous. Doesn't matter how many genes you're looking at. If it's a Mendelian cross, it is always homozygous parents, heterozygous F1, and then a whole bunch of different things in the F2. And so the heterozygous always expresses the dominant phenotype or phenotypes because it has a dominant allele for each gene. So yeah, the people that said dominant is correct because what you're looking for is what is the dominant phenotype in the F1. That's the dominant phenotype. Now the people who said F2 are also correct, right? Just will take a little bit longer. But in the F2, you'd expect a particular ratio, and the ones that you have the most of, which is the 9 of the 16th, will be dominant for both of those. So that's correct as well. That's another way of looking at it, which I hadn't thought about before. Okay, so we know from looking at where we at? The F1 generation that it's heterozygous, you see, for both of those, and it's round and yellow. So when you look at the P generation, P1s, or P0, some people call them, you see that this is the dominant, these are the dominant phenotypes of those 2 traits. Okay, now I'm going to draw this out because I'm not a big fan of trying to describe visual things with words. It doesn't ever seem to work super well. So I probably better get up now because I'm going to need to use the whole board. Ooh, making bones. All right. Okie doke. So here's our dihybrid cross. If you ever point the laptop in like some funky thing and you're just looking at the ceiling, let me know and I'll point it back in the right direction. So again, we have P1, F2, right? We have 3 different generations, just as we do for a monohybrid cross, exactly the same, never changes if it's a dihybrid cross, right? So you guaranteed, I could give you just the F1s. Sometimes I do that in exam questions, like blah, 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 blah. I have this particular phenotypes. What are the genotypes of the parents? Easy, right? You just work backwards because you know exactly what they're going to be. Work downwards into the F2s because you know exactly what they're going to be. So let's use, which way around was it? I think it was round first here. So we're going to have one parent, homozygous dominant for both, versus the other parent, which is homozygous recessive for both. Now, it doesn't have to be that one parent is dominant for both. As long as you have one parent that's homozygous dominant for that gene and the other one is homozygous recessive. I could, if I was particularly grumpy and feeling really mean, you don't have to copy this down. I'm just using it as an illustration. You could also do that as well. Don't really tend to do that very often, I think, by convention, and also because we don't want to confuse our students any more than they're already confused. So by convention, we always put it that one individual is dominant for both and the other one is recessive for both. But it doesn't have to be. And really, in the end, it doesn't make any difference anyway, because whichever way around you have it, you're always going to get heterozygous F1s. So next step, and I've taught this stuff for years now, right? So I kind of have a fairly good sense of where people get tripped up. One of them is students tend to lose genes, right? You have to remember that only one allele from each gene is going into a gamete. But both genes will be present in that gamete. So in the parents, you're going to have 2 of each letter. In the gametes, you're going to have 1 of each letter. But you are going to have each letter present. So that being said, what are the gametes we're going to get from this parent? Yeah, RY, exactly. And over here, yeah, it's also going to be RY. It's just going to be small ones, right? And you'll see the reason why I kind of been banging on about this when we get to the F2s. So those combine together, and now we get our heterozygous F1s. You always put the alleles, different alleles of a gene together. So it wouldn't be RY, RY, it's always RR, YY. These are always heterozygous. F1s are always heterozygous if it's a mono or dihybrid cross. Okay, now as it said, shit gets real. What gametes can we get from that individual? Have a think. What are the different possibilities? So remember, you're going to have, bloody hell, Carissa, anything. Michael, that's not sufficiently precise. Sorry. I feel a little bit more, yeah, this is genetics after all. All right. So Carissa got it. Jess, that was a great example of what I mean by losing a gene. So gametes are always haploid, right? Because if you think about it, if you've got two alleles in your own somatic cells, eyeballs, skin, whatever, then the only way to get two is to add 1 and 1 together, right? It's like the simplest form of math you can get. So haploid, the gametes have to be haploid. They can only have one allele of each gene, because when they come together, right? So these are essentially, female and male, or male and female, doesn't really matter. When these come together, now you have the diploid offspring, diploid individual. Right, that's a key part of sexual reproduction, is that you have to go through a haploid stage to end up again with diploid offspring. Right, so that was great. Don't feel bad about that, because that's a good thing to make a mistake in now so that we can correct that. So what you wrote was essentially a diploid gamete, which you can't have. And you also lost the Y gene, right? So keep track of that. So Clarissa got it right. So essentially, and the way that you can write it down is, well, actually several different ways, right? You can have either this or this, right? So you can have capital R or lowercase r. And the reason why I've written it out twice is whenever you get a capital R, you could get either a capital Y or a lowercase y. Similarly, whenever you get a lowercase r, you could have a uppercase Y or lowercase y. Right? Because you have two different genes. And if you have two different alleles for two different genes, you have 2 by 2 different options, 4. Right? So when you have two genes, you have, and each with obviously two alleles, you have 4 possible gametes. This is where stuff gets complicated. So for each of those, you either have a dominant Y or recessive Y. Dominant Y or recessive Y. These are your 4 different possible gametes. Now, again, I'm not saying that those are necessarily going to be taking part in that fertilization, but they are gametes that could be present. Okay, so law of segregation is about what? Questions to the class. What is the Mendel's law of segregation all about? What does it refer to? A little bit of revision for you. Yeah, Mika, exactly that. Good guess, Michael. But it applies to well, the monohybrid cross is what illustrates the law of segregation. So you're right on that point, Michael. But Mika is right. Alleles segregate into gametes randomly. Yeah, and that's involved in cell division too, Destiny, for sure. So if you have two different alleles, which is essentially the F1s of a monohybrid cross, then you have a 50-50 chance of getting the dominant or the recessive allele in the gamete, right? That also applies to a dihybrid cross. But now we have an extra wrinkle. That extra wrinkle is now we have two different genes that we're working with, right? So for a dihybrid cross, now we have the law of independent assortment. And what that is all about is saying that what allele you get for the, what is it, seed shape gene, right? Could be this or this, dominant or recessive, has absolutely no effect on which allele you get for the seed color gene, which can be either dominant or recessive. These two genes, or really these two genes, act independently. They're sort independently into these gametes, right? So they do not interact, they do not affect each other. I mean, that's the law of independent assortment. And the dihybrid cross really illustrates that very nicely. Okay, so that said, given that alleles segregate randomly, and the allele for one gene has nothing to do with the segregation allele for another gene, what ratio would you expect to find these gametes in? These four different possibilities. Think back to what we did at the test cross and with the F1s of the monohybrid cross. Same principles apply. Almost, Mika, but how many different possibilities do we have? We don't have just two, right? Yeah, exactly. We have four. Cool. So this is going to be in a 1-to-1-to-1-to-1 ratio. If the law of independent assortment holds true for these two genes, right? That's one of our assumptions. You can actually use this type of cross to test that assumption. Okay, so for our F2s, what we're going to be doing is crossing two F1s together. They're going to be selfed, right? So the progeny of the P1 cross, F1s, are going to be crossed to themselves, right? It's called selfing. Right, and so I'm just going to move this over here. Actually, ooh, that's ugly. I'm going to put another F1 over here. Right, let's tilt it down a little bit. And this F1 produces the same gametes as this F1. They're exactly the same. So we're going to have the same four possibilities. Like so. Now you can start seeing the outlines of that Punnett square that you get are 9-to-3 to 3-to-1 ratio from. And so let's draw. It's so much nicer doing it on this big whiteboard. Draw some nice lines. Like so. And then all you do is you fill in the squares. It's really quite straightforward. And if I had a third arm, I'd use my fingers, right? I can do it on this one because it's really close together. Normally I use my other hand as well. So you just add those together. It's going to be homozygous dominant for both of those. And as I do it, I'd suggest you do it too. Actually, you can do it faster than I do if you want, but just add these together, right? Don't jump ahead. You have to really, and this is what I do too when I do this. I work off what's in front of me, right? Rather than just kind of assuming what's there. That's why it's useful to use your fingers. Right, so the first line should look something like that. Hopefully with better handwriting. And then you just do it for the second line. And for now, don't worry about the genotypes and the phenotypes. We're just writing things down. We'll figure out what the phenotypes are once we've finished. Oh, nearly jumped ahead on that one. And again, this is one of those things that if you do all this, it'll always be right. Right? Cannot get it wrong. And then the very last one will be your double homozygous recessive. Okay, so what you have should look exactly the same. If you put the gametes down in the same way that I did, you'll get exactly the same Punnett square. If you swap these around, that's fine. Doesn't matter. You'll still get the same outcome. It will just look a little bit different. Has everybody had a chance to do this? Cool. And again, that's the thing with like Mendelian genetics is it seems complicated, but once you figure out the rules that it's working to, it's really quite straightforward. Right? It's just fairly, in a sense, fairly basic logic. So now what you do, we're not going to be doing a genotype ratio for the dihybrid cross because that would just be painful, right? What we care about are the phenotypes. And so now what you do is you go through all of these and you see where do I have a dominant allele, right? And so first we care about where do we have a dominant allele for both genes, right? This is one that's these are individuals that will express both dominant phenotypes, right? For each different trait. So the round yellow peas essentially. So we have, and it doesn't matter whether there are 2 dominant alleles or 1. Again, you will still get the dominant phenotype because that's the point. So you just go through, you can circle them if you want. You just count them, doesn't really matter. and just read off the phenotypes. Yeah, there we go. So all of those are dominant. So if we were to write out the phenotypes here, I got a bit of reflection from the window. You should get 1, 2, 3, 4, 5, 6, 7, 8, 9. Now, when you're looking at the parents, right, which are dominant, recessive, right? So we've got our round yellow peas. How many of the wrinkled green peas do we have? Well, it's just one down here. Almost, Jasmine. Almost. Just missing a number there. So this is wrinkled and green will be 1. Yeah, there you go, Jasmine. That's right. Let me just grab a different color. Purple. Who does not like purple? Oh, most people actually. But anyway. So now we're going to get 2 other types, which are kind of essentially a mix of the original parental phenotypes. So these are individuals that we have not seen so far, because we've either had both dominant, both recessive, or both dominant phenotypes. Now, because these 2 genes are sought independently, now we can get different combinations in the offspring. And this is what we're going to see now. Those are the 3-to-3 individuals. So now we're looking for, what would that be? That would be round and green. It's going to be, so we're looking for a dominant R and a homozygous recessive Y. So we have 1, 2, and 3. So that gets us our 3. Ooh, got green. And now we need to do the same for the other possible combination, right? Because if you have round and green, then you have to have wrinkled and yellow, right? They're reciprocals. You can only have one of them if you have the other one as well. Right, now we do the same. Now we're looking for homozygous recessive for R and a dominant allele for color for Y. We have 1 here, 1 here, and 1 here. Oops. That gets us our 9-to-3 to 3-to-1 ratio. And that is, crap, drop my pen lid. The only output that you'll get from a dihybrid cross. If these alleles are segregating randomly and the alleles for one gene have no effect on what alleles you get for the other gene. If those 2, and obviously the other kind of non-specific assumptions, no mutations, no selection, and random reproduction, this is what you'll get. So if you see a 9-to-3 to 1 ratio, like in the offspring, say you're going to have, I don't know, like 91, 28, 33, and 11 different phenotype combinations. So 9, it's dihybrid cross. Okay, let me just take pictures of that while you have a think. Also means I can put my laptop down. Might not weigh much, but when you hold on to it for a while, it does. Yeah. Coffee. All right, questions. Gotcha. Okay, so practice always makes perfect, as they say. So let's do this cross. Always have to be a little careful. I don't want to offend anyone who's short, but I always like using dwarfs because I think of Snow White and the Seven Dwarfs for some reason. So with what would be the opposite? I don't know, happy non-dwarfs, I guess. Okay, so there's your P1 generation. You get a bunch of grumpy dwarfs. That's the F1. So two questions. Question one, what are the dominant phenotypes? Should be fairly straightforward. Question two, what offspring and ratio do you get in the F2 generation? Okay. While you do that, I'm going to wipe this off. In a sense, you could actually just jump ahead and go, oh, it's going to be this, this, and this, and this. If you really understand it, I'd actually recommend doing it the long, laborious way because A, we've got time, and B, that's how you learn how you can make those shortcuts. So have a go at that, and I'll clean the board off while you do that. Give me a shout when you're done. We can go through it. Jess, you raised your hand, virtually speaking. Oh, okay. All right, I think most of you kind of starting to wrap up. So while you do that, I'm going to go over to the board and we can work through it, make sure everybody's kind of getting the same answer, which you should do, right? Let's go put it up. And this won't take very long to go through because we can skip a few of the explanations because we've already done that. So grumpy dwarfs, let's have, question 1. What are the dominant phenotypes? Grumpy and dwarfs. Good job, Krista. Got to be that because those are the phenotypes you see in the F1s, right? That's the easiest way to tell. So that'll do. So these are our, that's grumpy, not crumpy. Crossed with our, what is it, happy non-dwarfs. So again, right, have to use the same letters, just upper and lower cases. Can't invent new ones. So it wouldn't be H, for example, right, for happy. This would be our happy non-dwarfs. Snow White, I guess. Though whether she's happy or just asleep, it's kind of hard to tell. Okay, so we know that the F1s have to be heterozygous. Those are going to be that. This is our F1s. And we're going to have the same over here as well. Now, good practice again. What gametes can we get from these F1s, these double heterozygotes? What are the potential gametes? Yeah, good job, Mika. So it's going to be big, big. Yeah, that's good spacing. Big, little, little, big, little, little. Right? So you should have those four. And we're going to have the same again for the other individual because it has the same genotype. Like so. Draw out your Punnett square, and then you just fill them in. Right? I'm going to do a super quick. Speed crossing. And this should be exactly the same if you have the same order as I do that you have in front of you. And really the key to being quick at this stuff is practice. So you're going to get questions like this in the exam, guaranteed. You're not going to have extra time to do those questions. So you have to be able to do this fast. Or in some cases, just go, I know what the phenotypes for the dominant phenotypes are because I've been given that information. I just have to interpret it. All right, so read off. We're going to have these ones are going to be our grumpy dwarfs. This one down here is going to be our other parent. And I refer to these as, I call them parentals, right? Because those are the phenotype combinations that you see in the parents. We'll be using that language again when we talk about recombination, which is kind of how this gets broken, essentially, sort of. That's not a very good way of describing it. So happy non-dwarfs will only have one. And then we have grumpy non-dwarfs. We have 3. And then happy dwarves, which is what everybody wants, is also 3. Now, in a sense, right, you didn't even need to do all of that because you know that you're going to get a 9:3 to 3:1 ratio in the offspring of the F2. You know that you're going to get 9 of the dominant parent, 1 of the recessive parent, and 3 each of the reciprocal mix of those two. So you're going to have 3 grumpy non-dwarfs and 3 happy dwarfs, right? So in a sense, you don't need to do all of this, but you need to understand all of this to be able to do that kind of leap ahead, right? And you should all hopefully get the same. I'm going to take a quick picture because there's one last thing that I want to, if you didn't, that's cool, but let's figure out why. I just want to take some pictures and clear that off because there's one last thing that I want to do. Okay. Now, let's go back to our test cross. What about if you have an individual that is dominant for 2 traits? How do you figure out what the genotype of that individual is? Right, so this is a little bit more complex, right? Because we have more stuff going on. So let's say we have 2 genes. Let's go with the grumpy dwarf scenario. What would our test individual be? Right, it's got to be homozygous recessive for both. Correct. What's the phenotype of that, Clarissa? These are going to be our happy non-dwarfs. So this is a little bit more advanced, which is why I've kind of left it to the end. Now we have 1, 2, 3 possibilities. We have our homozygous dominant for both. What other possibilities do we have? That would be have dominant phenotypes for both. What are the other 2 possibilities in terms of genotype? Remember, you only need, actually, oh, there are 3 other possibilities. I'm not doing a good job here. I'll give you one. Right, could be heterozygous for both. Oh yeah, there you go, Chris. And Rachel and Lindsay as well. Good stuff. So you could be this. Shoot, I didn't leave myself enough space. Or that. So this might look like something that's just horrendous to try and figure out, right? Because now you've got 4 different potential possibilities with the same phenotypes. How do you figure out what the genotype are of your unknown, which could be one of these 4 things? You just do the same as we did before and figure out what alleles are you going to get, right? So we've already done this one. That's exactly the same as what we just did for our dihybrid cross. Now this isn't a dihybrid cross, it's a test cross, but we still have for a double heterozygote the same 4 potential gametes. We also know, I think it was Jesus, maybe someone else, you're going to find these in a 1-to-1 to 1-to-1 ratio, right? So what gametes can you get from this individual? What's the only possibility you can get? Great, Mika, you can get a double recessive, right? Lowercase g, lowercase d. So then you're going to have essentially 4 different possibilities. You're going to have the 4 different phenotype combinations, the same that we saw in dihybrid cross. But now, because there's only one possible gamete from the other parent, you're going to have a 1-to-1 to 1-to-1 ratio, which is the same as the 1-to-1 ratio you'd get from a 1-gene test cross. What are you going to get if your individual is homozygous dominant for both genes? You can unmute, Jess. I'm not that good at lip reading. what are you going to get in terms of progeny if your unknown is homozygous dominant for both? It's going to be 100% dominant. Okay, so that's easy. What are you going to get here? All right, this is kind of pushing you a little bit, but it's worth doing to kind of stretch your brains out a bit. Although the last minute of class might not be a good plan here. Kind of, Amanda, kind of. So what different gametes can you get from this individual? You can only get one type of gamete or one type of allele for this because it's homozygous dominant. What alleles could you get for the grumpy gene? Yeah, exactly. You could either get dominant or recessive. Yeah. So now you're going to get, or let's do 1-to-1 grumpy dwarves to happy dwarves. Because the only gene that's heterozygous is the grumpy gene. And just in time, you can work this out if you want for yourselves, but you get the opposite if it's heterozygous for the happiness gene. You'll get only grumpy individuals, but they'll either be happy or, hang on, no, only grumpy individuals, and they'll either be, crap, got this wrong, didn't I? You're probably looking like, what was he talking about? This is going to be, yeah, that's right. Yeah, I did get that right. Huh. Well, it's always worth checking. They're all going to be grumpy, and they're either going to be dwarfs or non-dwarfs. Right. So the exact same principles apply as with a single gene test cross, just as the same principles apply to a monohybrid cross as they do to a dihybrid cross. Now, I got to get this all picture taken of and rush over to lab because that starts in 14 minutes. So I'm going to sign off and catch you either in 13 minutes now, crap, or on Tuesday. So thanks everybody. Practice this stuff, okay? It's the only way you're going to get good and quick at it. See you anon.